Good day, everyone. Today we're going to discuss the biology part, which is our part one, our basic microscopy. So the first part of this lesson is about the microscope. And again, if you will be needing our topics for biology and the other topics for the other subject, you may want to visit this Google sheet. And so I listed all the possible lessons that you're going to need to cover from the biology. So as of now, we have the basic microscopy parts, and then the next would be the levels of organizations. Okay, so let's discuss now. So we all know the parts and function of a compound light microscope. I think you are discussing this since you were in grade 7 or in your elementary days. So the most important thing that you need to take note of this is the diaphragm, wherein it controls the amount of light passing through the slide. So now, take note that when using in microscope, we need to understand these terminologies. We have the magnification versus resolution. Magnification is how much bigger a sample appears to be under the microscope that is in real life. So the overall magnification will be the objective lens times the eyepiece lens. So here we have this objective lens. So we have here the eyepiece, and then here are the different objectives. So what you're gonna do here is to multiply the ocular lens or the eyepiece with the objectives for you to get the overall magnification. So we also have the terminology which is known as the resolution. It is the ability to distinguish 2 points on image. For example, the amount of detail. Take note that the amount of detail depends on the resolving power of the microscope, which is the smallest separation at which two separate objects can be distinguished. So usually, the resolving power of a microscope is limited by a wavelength of 400 to 600 nanometer for visible light. So the wavelength of light is much larger than the wavelength of electrons. So the resolution of the light microscope is a lot lower. So when using a microscope with a more powerful magnification, it does not mean it will increase the resolution. In fact, a more magnification will not increase the resolution any further than 200 nanometer if we are using the light microscope. Why? It will just increase the size of the image, but the objects closer than 200 nanometer will still only be seen as 1 point. So, by the way, there are two types of microscope that we need to understand. So, we have first the light microscopy or the light microscope, and then we also have the electron microscopy or the electron microscope. Now, there are some points that you need to take note when using this. We are familiar with our school, which is the compound microscope. This is the one that we are using in our school. And light microscopy is the oldest, simplest, and most widely used form of microscopy. Specimens are illuminated with light. So we just focus using glass lens and viewed using the eye or photographic field. So there are different type of light microscopy because again they use light. We have here the fluorescence microscopy aside from compound microscope, and we also have the interference microscopy. Now the compound microscope that we are using in our school, the resolution must be greater than 200 nanometer. So if it is lower than 200 nanometer, it will be difficult for us to visualize the object. So in this case, this is the sample of fluorescence microscopy, and then this one is the uh, interference microscopy. Now take note that we don't see the details of this object. Okay, for example, that we don't see the details of the organelles of the cells. We only see the cells. Of course, some of the parts of the cells, yes, but the details of the organelles, it's very difficult to see. Now, when we want to do that, we now use the electron microscopy. However, there will be a problem because by viewing that, there must be we cannot use light, okay? Because light is it has a greater wavelength as compared to electrons. And remember, light, it has a 400 to 600 nanometer for visible light. Now, if we want to have a more detailed one, we will use electron microscopy, wherein it uses a beam of electrons rather than electromagnetic radiation to eliminate the specimen. Electrons behave like waves and can be easily produced using a hot wire, focused using electromagnets, and detected using a phosphor screen or photographic film. And then a beam of electron has an effective wavelength of less than 1 nanometer. So with this invention, during the 1930s, it revolutionized biology simply because it allowed organelles such as the mitochondria, the endoplasmic reticulum, and membranes to be seen in the details for the first time. So this image is an image coming from a electron microscope. Okay, so we have here the uh, transmission electron microscope. And so here is a detail of the mitochondria. So we also have two types of electron microscopy. We have the transmission electron microscope, which works like light of microscope, transmitting a beam of electrons through a thin specimen and then focusing the electron to form an image on a screen or on film. Okay, we also have the scanning electron microscope. So this one has scans a fine beam of electron into a specimen and collect the electrons scattered by the surface. This is poor resolution, but this we're gonna give an excellent three-dimensional image of surfaces. Okay, so again, I want to emphasize that for electron microscopy, uh, it is more detailed and it is less than 1 nanometer, and it will be used for the details when viewing the organelles of the cell. Now take note that if we are using electron microscopy, the specimen cannot be alive. So specimen must be fixed in plastic and viewed in vacuum and must therefore be dead. Aside from that, they are using heavy metals for staining or for visualizing. Okay, so next it's also important to discuss how to use a compound microscope. One important thing when we are using a compound microscope, in using a microscope, adjust first the LPO before shifting to adjust the HPO. It's always the LPO first, so the low power objective first before the HPO. You can also watch a video in YouTube how to use, but what I just want to emphasize here that LPO first before the HPO. Another thing here is that when we are using LPO, we can do the course adjustment knob, and then never use the course adjustment knob if we are in HPO. Okay, so the course adjustment knob, that would be for LPO, while for the fine adjustment knob, it can be better used for the HPO. So take note that the formula for magnification is the size of the image over the size of the actual object. So there are some conversion factors that we need to know. For every 1 millimeter, that is 1,000 micrometer. 1 centimeter is equal to 10 millimeter. Take note of that. And 1 micrometer is equals to 1,000 nanometer. So let's try to look at some questions regarding this. Question number 1. The eyepiece has a total magnification of 10 times and the LPO was a magnification of 40 times. What is the magnification or the total magnification? Of course, this is just simply multiplying 10 times, 40 times, and that would be letter uh, C, that is 400 times. Okay, I think I write the solution here also. So, so this one is letter C, 400 times. Next, number 2. In light microscope, sometimes a blue light is used to visualize the image better. What could be the possible reason for this? Now, take note that the answer here is that blue light has short wavelengths and thus can increase resolutions. So for here, let's try to look. Blue light increase the magnification. No, we don't increase the magnification. We are increasing the resolution. Blue light has greater wavelength compared to other light, which is around 600 nanometer. That increase the resolution of the image. So is it greater wavelength? So it's not greater wavelength, but rather blue light has short wavelength and thus increasing the resolution. Okay, so How about letter D? Blue has greater wavelength than electrons wave and can increase the resolution power. Okay, but we are not using, but electrons wave is not related here, right? So letter D is not the best option. How about number 3? Which of these is true about microscopy? So here, is it A, compound microscope like fluorescent microscopy can visualize the details of mitochondria? No, we cannot visualize the details of the mitochondria because we need to use the electron microscopy for this. Okay, electron microscopy. Now, how about the other one? Electron microscope used to visualize small object less than 1 nanometer. Next, letter C. Electron microscope can visualize live cells and movement of the detailed cell parts. This one is wrong. Letter D, increasing the magnification of a compound microscope will increase the resolution of the image. No, it doesn't mean that if you're going to increase the magnification, it will also increase the resolution of the image. So it's not related. So letter B, electron microscope used to visualize small objects less than 1 nanometer. Okay, so next, how about number 4? An image of a cell has a diameter of 8 millimeter, while the actual size is 20 micromillimeter. What is the magnification? Now take note here that what is the magnification out of this? Now take note that uh we can only we can only solve for it if we have the same uh, unit. That's why we need to convert this 8 millimeter to micrometer. So we have your 8 millimeter and then times 1,000. And remember that when we use dimensional analysis, okay, just want to recall this part. Is 1 millimeter is equals to 1,000 micrometer. If you are still confused out of this, take note that the 1 millimeter is placed at the bottom part of the dimension and 1,000 micrometers on the upper part because you want to cancel out the millimeter. And so you will be left out with 8,000 micrometer. And take note that the magnification is size of the image form over size of the actual object. You don't need actually to memorize the formula, but rather understand the formula. Why is it like that? Okay, and so that is uh 8,000 over 20 micrometers. So if you are still confused while doing this, so cancel out the 0 and 800 divided by 2, and that would be 400 times. Okay, so the magnification is 400 times. Now, how about number 5? An image is 4 centimeter wide when viewed under 400 times magnification. What is the actual size of the image? Now, first, so we have your 4 centimeter, and then we have 400 times magnification. Now, if you're going to look the choices, it's all of these are micrometers. And so we can simply, again, convert it into micrometers. So 4 centimeter. Now, take note that we have 1 centimeter and then 10 millimeter. I can simply cancel that out. And so that would be 40 millimeter. Now, since we want it to convert into micrometer, we can continue now. We have 1 millimeter and then we have 1,000 micrometer. And so cancel out the unit. And so overall, that would be 40,000 micrometer. So this 40,000 micrometer, that's the image is 40,000 micrometer. So what is the actual size of the image? Now take note that we have here the magnification size or the magnification formula. That would be magnification, the size of the object form and size of the actual object. So that would be M. You can try to write the formula first. So magnification is size of the image over size of the object. Now, if you want to derive it using algebra, of course, we're going to have O and then I over M. Okay, you just simply, again, remember that we multiply this by O, and that could be OM, and that would be I, O will be canceled, so this one is I, and since we are looking now for O, that is I over M. And so what is now our formula? The size of the actual object equals the size of the image form over magnification. So by doing that, that would be 40,000 micrometer divided by 400. Now you can see here that I can cancel out the two zeros here, right? And so that would be 400 over 4. That is 100. And so that would be 100 micrometer. And so that is now our answer for number 5, which is letter D, 100 micrometer. Okay, and that's all. You may now try the practice set number 1. You can pause the video. So we have here the number 1, number 2. We also have question number 3 and number 4. And then we also have question number 5. You can pause and take a screenshot of the video and try to answer it. And explanation will be on the other part of the video. Thank you very much for listening.